expertise and to give insight of our students here and our colleagues. And for this session, we will start this MRT by having some opening remarks from our colleagues here on behalf of the head of the school, from Dr. Fidelis Niti. Time is yours, Pak Fidel.
Good morning, everyone. Thank you very much, Pak Rainer, for giving me the opportunity to deliver an opening remarks for this morning's MRT monthly research talk. So, good morning. Today we have our special guest, Mr. Mia Haruni, who is a student at. Am I audible enough now?
Yes,
Sorry.
yes.
All right. Okay. Um, so good morning to our special guest today, our special speaker today, Mr. Mia Haruni who is a PhD student at the Faculty of Industrial Sciences and Technology at UMPSA, Malaysia. So welcome to this online monthly research talk. So this is our regular talk, which is conducted every month, where we invited special guests from around the university, both in Indonesia or overseas, to give their insight or to update us with their current research. So this morning we have here Mr. Mia Rooney to deliver a talk on the importance of in silico study in drug discovery. So on behalf of the um, head of department, the coordinator of the chemistry department at the Faculty of Science and Engineering at the University of Nusa Cendana, we would like to extend our heartfelt grateful for your presence today in the MRT. We hope that this MRT will go smoothly and we'll have a new insight from you, Mr. Miaroni, especially about the importance of in silico study in drug discovery. So we also have here not only our lecturers but all our students. So we hope that this today's MRT will uh, deepen their understanding and give their new insight about in silico study in drug discovery. I think that's all from me. I handed it back to you, Prof. Reiner. Thank you very much.
Thank you very much, Fidel. Now time for Mr. Rooney for delivering his talk about doing in silico studies, as he is expert in that field. He published many papers, and when I was in Malaysia, his colleagues said that he is the most productive man of producing high quality papers of the university. So, Mr. Rooney, time is yours.
Thank you very much for giving me this opportunity. Are you seeing my slide?
It's opening now, but we haven't seen the slide yet. Okay.
Assalamu alaikum and very good morning, everyone. This is Miaroni, a PhD candidate in chemistry in University Malaysia, Pahang, Al Sultan Abdullah. With the thanks to the Almighty Allah and my supervisor and co-supervisor, I would like to share with you about an interesting topic, the importance of in silico studies in drug discovery. We are affected by disease and we need drug to cure to them. So we need drug every day. And need to drug discovery, need to need to discover a new drug. It is an ongoing research. By the way, conventional drug discovery process is time consuming and costly. According to literature, it needed about 1 to 2 billion US dollar, and about 8 to 10 years are needed to discover a new drug. And in conventional drug discovery approaches, we get some biohazard, which is harmful for the health, for the environment, as well as for the animal. On the other hand, computer-aided drug discovery, I mean in silico approaches, is time safely and cost effective. Using computational approaches, we can easily find out the lead compound, and we can reduce the time and cost. What is in silico approaches? In silico approaches is the method where we used computational simulation and artificial intelligence to design, to find out a drug candidate, which is reduce the time than the conventional drug discovery approaches. We can easily design a protein structure for a disease using in silico approaches, and as well as we can screen a big library, big compounds library within a minutes or very few time. So it helps us to reduce the time, reduce the cost of conventional drug discovery or development process. Conventional drug development process have a lots of I mean a conventional drug development process is time consuming and costly. On the other hand, in silico approaches, I mean computational simulation based approaches have several advantages, including cost effective, time consuming. We can approach for experimental approach from the conventional drug, from the computational drug discovery. It supports the rational drug design easily. Um, and In conventional drug development approaches, if we want to find out the toxicity or absorption, metabolism, it takes 1 to 2 years or more than 5 to 10 years. But using in silico approaches or computational approaches, we can easily, within a seconds, we can find out the properties of a compound, physical and pharmacological properties of a compound within very few times. Although in silico approaches have several advantages, but it has some disadvantages, including still now we are depending on model accuracy, quality of data, and for some experimental the process are not fully captured. Still we need wet lab validation, and also software and computer price. It is these are the disadvantages of in silico approaches. I explained in previous slide that using computational simulation and artificial intelligence, we can design and identify a lead compound that's called in silico approaches, which is covered admit prediction, drug likeness, virtual screening, molecular docking, molecular dynamic simulation, network pharmacology or system biology, and then density functional theory. So now I would like to share with you step by step. So what is ADMIT? ADMIT is the short form of absorption, distribution, metabolism, excretion, and toxicity. We can determine the drug safety and efficacy of a lead compound or a compound using this approach. So how will we know which compound is better? Suppose we are trying to screening a big database where a million compounds are there. So which one is our lead compound? Which one or which two? Which five? Which 10? It's depends on some value and some methods. Like for absorption, water solubility. How was the water solubility of that or those compounds? How about the blood-brain barrier? I mean distribution. Then how was the metabolism, excretion? How was the toxicity? Based on these parameters, we can select the best compounds. So what is absorption? Absorption is the first process of a drug. Using this process, our body or our skin, our gut, human intestine absorb a compound or a drug. We can determine this using some computational simulation based software like PKCSM, SwissADME, then admit lab, etc. So poor compounds are always elimination from the database and which absorbed by the human intestine very high, that is selected for further step. And then distribution, unmetabolized drug transferred to the blood stream or tissue. Using this process, we can volumetric analysis for blood-brain barrier, and using this process, we can determine the dose of a drug. To predict this, we can use the previous software like PKCSM, SwissADME, ADMIT-Lab. Metabolism, how a drug is breakdown in our gut or in bloodstream, it is determined by metabolism. Usually in our body, more than 70 CYP, cytochrome P450 isoform or inhibitor are existing. Among them, one cytochrome that's called CYP3A4, which is responsible for 90% metabolism of a drug. Metabolism helps us to identify a lead molecule and drug-drug interaction and toxicity and calculate the dose also. Then excretion, it is the last step of a drug. So how a drug excretion from elimination from the body is called excretion, which is kidney is the responsible for drug excretion, and using this we can determine the half-life of a drug and how was uh, how was the duration of action of a drug, we can determine using this. And then the last of admit is toxicity. Using these parameters, we can determine the toxicity of a compound using several software like Protox, AdmitLab, then PKCSM. Like suppose a drug is toxic, so it is not good for human body, like hepatotoxicity, cardiotoxicity, mutagenicity, or lethal dose 50. It is not good for health. But we can determine the dose or we can determine the toxicity to um, which amount of toxicity are consuming by our body, we can determine using this. It have also some parameters. Here I have added a figure where I tried to show the toxicity of a compound to accept as a drug, like very low, less than 10. It is possible to non-toxic, and then 20, and high and extreme high are not good as a drug. So when we screen of a database, we should exclude them. Drug likeness is referred to a molecule when it followed when it follows the rule to be a drug, and which is have the similarity of existing drug. So how will we determine that it will be a drug? So we have several rules. Among them, Lipinski's rule of 5, that contains molecular weight should be less than or equal 500. Then hydrogen bond acceptor should be less than or equal 10. Hydrogen bond donor less than or equal 5. And Mi log P value less than or equal 5. Um, uh, um, Without this, we have another method which is invention by Weber, Gauss, Egan, Mach. This is also for shows a compound will be a drug or not. Then virtual screening approaches. Using a database, suppose a database contains 500 compounds. So we want to discover a compound, a lead compound against a disease. So how will we choose from 500 compounds? If we go for conventional drug discovery approaches, then we need year after year to select the best compound using in vitro, in vivo, I mean in cell or in animal. So, but using the computational simulation-based study, using this approach, I mean virtual screening approach, we can identify within a day or within a minute, like ADMIT study, then molecular docking, molecular dynamic simulation to find out the best compounds. I already explained the acceptance range of all parameters. Using that, we can virtually screen, or we can eliminate the existing which doesn't follow the rule, and which follow the rule, we can choose them. It is called actually screening. Um, to source best compounds from million compounds for hundred compounds. Actually, it is called virtual screening. Here I have added a figure from one of my study. There I arrange a in-house database which contains 130 compounds. Then I perform pharmacophore based screening and I found 40. From then I perform molecular docking study to find out the heat compounds. I found 11, of which admit and drug likeness filtering I found 3. Then I perform the lethal dose 50 toxicity, mutagenicity, carcinogenicity, hepatotoxicity. Then I found only one compound as lead, which then for which I perform molecular dynamic simulation and propose that is the lead compound from 130. So this is called virtual screening. Then molecular docking. Molecular docking actually is the main part to discover a drug using in silico approaches. Why I called it main part? Because using this we can know how a compound bind with a protein of a disease. Um, shortly, docking means parking. Like I need to park a car in a specific area, I mean parking area, and there have a spot or line to park my car. That parking spot is the active site of a protein, and ligand compound is my car. So I need to park my car in that parking area. So I need to bind the ligand in the specific binding site of a protein. That is the docking. Docking is the binding mode of protein and ligand. How a ligand is bind with the protein. It is called docking. Using the docking and docking energy from the docking, we can get the binding energy. Using that, we can say that which compound is active against that disease. So from molecular docking, we can find out the lead compound, and to determine or to perform the docking, we have several software, including Autodoc. It's free. CBDoc is online based, free. Schrodinger, Discovery Studio, it's paid, but considered as good, and all of the software are supported and accepted by the scientific authority. Then here I have added the workflow of docking. First of all, we need to select the target. Which one will be target? Suppose I want to discover the drug against COVID-19. So I need to select a protein, COVID-19 or dengue virus. Dengue virus have 4 serotypes: dengue 1, 2, 3, and 4. And have 10 proteins, 3 structural and 7 non-structural. So which one will be the target? So I need to choose the best target, and then I need to prepare the protein, then prepare the ligand. Suppose I want to perform the docking for 100 compounds, or in previous slide in this slide, I prepared 130 against dengue 3 RNA dependent RNA polymerase protein. So I need to preparation these compounds as ligand. Then we will perform the docking using software like CBDoc2, Autodoc Bina, Discovery Studio. Then we need to analyze to see the interaction, where the ligand is binded, or which amino acid is responsible to bind that, using various bond type like hydrogen bond, carbon hydrogen bond, alkyl, pi alkyl, and we need to see the score and compare these results with a standard compound. Standard compound, I mean, it will be a drug or native ligands of that protein, or which compound showed best activity in previously. We can choose them as control compound or standard compound, or we can call it reference compound. Here I have added some figures to explain you about the interaction. See here, van der Waals. These are the van der Waals which have no connection, no dot line like this, no dot line with that ligand. See, this is the protein. This is the protein, and this is binded ligand. This one. This one here actually bind with the ligand and amino acid residue, where here this is 3D structure and this is 2D structure. Using this 2D structure, we can analyze easily. See, this is van der Waals, and then conventional hydrogen bond, carbon hydrogen bond. Conventional hydrogen bond. See, the color is green. Here, a green dot line. Green dot line is means the conventional hydrogen bonds, and carbon hydrogen bonds is not fully green but greenish. So this one and this see, this one and this one have difference. So this one is carbon hydrogen bond, which is also one kind of hydrogen bonds. And then pi anion, this is pi anion, and pi alkyl, this is pi alkyl. Pi alkyl and pi anion bond is strong than the hydrogen bond. But if you want to find out a lead compound, besides binding energy, you need to see the hydrogen bond. How many hydrogen bond are there occurred? Based on this, we can choose a compound is active or non-active. After that, from this, this is the complex of protein and ligand. So we already performed the molecular docking simulation. So we need to see the stability. We need to see the accuracy. We need to see the stability of the docking complex. Suppose I perform a docking but not stable. I want to discover a antiviral drug, but I perform the docking that is not stable. So after taking that drug, we will not get the good results. So we need to check the stability of that docking complex. Using molecular dynamic simulation, we can perform the stability test of docking complex. And also in docking complex, when we prepare the protein or ligand, we need to eliminate the water molecule. In our human body, we have water molecule, but using docking, we need to eliminate them. But in molecular dynamic simulation, we need to add water molecule also, need to add temperature. In docking, we can't add temperature, but in molecular dynamic simulation, we will have to add temperature like human 37 plus 345 310 Kelvin. So we need to add a temperature. We need to maintain the human condition during molecular dynamic simulation. After performing molecular dynamic simulation, we will get RMSD, root mean square deviation, RMSF, root mean square fluctuation, then a radius of gyration, and interaction, as well as we will find the binding energy again. Right now, docking is not enough to bind a compound with a protein of a disease. So MD simulation is the best way to get the binding energy now. So if RMSD value less than 2, then our our complex is stable. Our complex showed stability when the RMSD value is less than 2. RMSF value is less than 2. RMSF value if less than 2, then our complex is stable. If more, then also we can consider it as stable when the apoprotein, unbound protein, only protein. During the MD simulation, we actually need to perform the perform for apoprotein. I meant only protein. Suppose I choose in previous study dengue 3 RDRP. So I need to perform the MD simulation only RDRP. Then which compound I propose as lead compound, that complex, and reference compound complex. Suppose apoprotein or unbound protein showed the RMSD value less than 2. then our compound if should less than 2, then we can say it is stable. Or the apoprotein should 10 angstrom, then our compound should 8 angstrom. Fortunately, our compound reduced the RMSD value. Then we can say that our compound reduced the RMSD value of apoprotein. Then our compound showed stability. As well as a molecular MMGBSA, this is one kind of binding energy we also get from docking, but it is best than that. It is the validation of binding energy. Binding energy we need, because without docking we can't perform molecular dynamic simulation. For molecular dynamic simulation we also need the complex, and MMGBSA also from complex. It is one kind of binding energy which is the validation part of docking. When it showed the minus energy, then our docking is okay. And based on the RMSD, RMSF, radius of gyration, hydrogen bond, SASA, secondary structure analysis, principal component analysis, then a free energy landscape, we can say that our compound is stable or not. And using MMGBSA approach, we can also say that our compound is perfect to approach as lead. Then it is the case study. I published it in 2023. The title was Identification of Pyrazole Derivatives of Eosinic Acid as Novel Inhibitor of SARS-CoV-2 Main Protease through the virtual screening approaches. In this study, we used, I forgot how many compounds we used, maybe more than 30. Pyrazole derivative, sorry, more than 50 more than 50 compounds. From the 50 we find out 2 lead compounds, and from the docking study we find out 2 which were then perform MD simulation. We should see the RMSD value and RMSF value very less than 2 angstrom. And finally, the interesting topics is network pharmacology. Prof. Lerick was here to learn especially this. This is the interesting study, I said, because from this study we can target gene to gene of a disease, protein to protein of a disease. That's why it's called polypharmacology or system biology. Suppose we have a compound and we want to discover a drug against a disease. With a compound, we can target multiple protein, multiple gene using network pharmacology. In another process, suppose docking, we can target only one disease in one time. But using network pharmacology, we can source multiple target. How? Let's see. Suppose we have a compound and we found a compound related from a compound related database, target around 453, and here I have added asthma related gene around 1417. Then we perform the overlapping to find out the common gene from this compound related and from this disease related. How many are common? So we found 177. Using this 177, we go for another software called David, called string software. Using that string, we can perform protein-protein interaction. Suppose here 177 gene targets around 200 protein, and that have interaction, obviously have interaction, we need to find out that using network pharmacology study. From this 177, we also get the biological process, then cellular component, molecular function. Cellular component, molecular function, biological process, everything we can get using network pharmacology study. And then finally, see this figure here from, sorry, excuse me. From that 200, suppose we found 200 protein-protein interaction, and from that 200, we find out 10. Based on the color, we can choose the multiple protein against a disease. In this study, asthma related study, here TNF alpha, then AKT1, MAPK3, these are the best based on the color. Which color is red is higher, and then rest of others. So this is the best target for asthma related target. See, TNF alpha is inflammation related. AKT1, MAPK3 is cancer related. For asthma, we also target inflammation and cancer related. So this help in several diseases to discover a drug. And we can find out a pathway, signaling pathway. Which pathway is better for that compound? Using the network pharmacology, we can easily find out the pathway. So which pathway is better and which pathway is best responsible for that disease, and using that pathway, how will we discover a drug? It is easily we can learn from network pharmacology. Using a compound, we can get the protein-protein interaction, then pathway enrichment analysis, mechanism of action using network pharmacology. Here, according to literature, to get the mechanism of study of a compound in conventional way, we need year after year only find out the mechanism, how a compound and which pathway is responsible, or which pathway is better for that compound. It's need year after year. But using network pharmacology, we can find out within a day what will the possible pathway for that compound against that disease. It is very easy to find out using network pharmacology, and we need several software like eString, David, then Cytoscape, and need several database like compound related target database, Swiss target prediction, then disease related database, DiseNet, OMIM database, then ZinCard database. So what is the benefit of network pharmacology? I already explained. Using network pharmacology, we can find out the possible pathway, find out the mechanism, and we can perform multi-target based drug discovery for a disease. We target a disease, but we can find out the drug for several diseases within a time. So from today's lecture, we already learned that the in silico approach, including admit, molecular docking, molecular dynamic simulation, and network pharmacology. Based on this analysis and according to literature, this approach help us to reduce the time of a study, to find out the lead compound within a few times, and improves the success rate. Um, it was for the day, Alhamdulillah, and thank you very much. And I would like to acknowledge to Dr. Larrick for giving me this opportunity to um, share my research with you all. Thank you very much. And if you have any question, please forward.
Right. Thank you very much, Rooney. That was a very nice talk, and it's very insightful for our students. For just for your information, before we going to the Q&A, there are about 39 participants in this for this morning MRT, and our students also have computational chemistry. They have studied computational chemistry, but in basic things that
MashaAllah.
I think what you delivered today could enrich more for them in doing, maybe to interest them with the computational chemistry.
Inshallah,
And
inshallah.
in chat room, we already have one questions for our from our alumni. He is also a lecturer and doing computational chemistry, and his name is Putra, and he asked several questions at the
Yeah.
same time, so that we start one by one. The first question was the prospect of in silico research. What is the prospect of in silico research for drug discovery purposes? The prospect for the future might be getting job by doing the computational chemistry.
Thank you for your very good question. And the last year Nobel Prize has gone for physics and chemistry in silico study, and it is right now uh, actually it is easy to invention and discovery new things without wet lab work. So I hope this will be very good in future. And already in clinical study has been approved, and already started about in clinical study. So using this in silico approaches, we can perform in clinical. Now imagine in future it will be a very good high, inshallah. And for job facility as a computational chemist and in silico studies, it will be open a new door, not new, already opened, a new insight for in future, inshallah.
Right. Thank you, Rene. The second question is about still for the future, as we already know that in the past, like me, what we studied just to modeling the molecules, and then through the improvement of computer specification. Right now we have internet, we have AI.
Mm.
Do you think that in the future might be the AI could, yeah, we can generate docking through the EA, just asking the chat GPT, could you do the docking or something like that in the future? Your prediction for that?
Yeah, for my study, here I will take a case study. For my study, I asked to ChatGPT to make a model of protein. And the ChatGPT prepared very well when I analysis using my software. That's is acceptable. That is very good and correct structure. So AI is for the health of human being, but we need to aware about that to use within ethics. That's all.
And the last question from Mr. Putra. Uh, when publishing the results of the research, when publishing the results of the computational chemistry, is wet lab confirmation required?
Uh, to publish a research, you can recommend for future. And until now, experimental validation is required. But if you for publication, if you add in the future recommendation part, it will be easily accepted by the journal and by the scientific authority, because our research followed by the industry, followed by the scientific authority, and they obviously ongoing on that. In future, if you recommend for future, someone will perform, inshallah. So it is with recommendation for future perspective, it is accepted.
Right. Left one. If it is still acceptable without wet lab confirmation, what parameters are still considered valid for reputable journal? If we just publish without the wet lab confirmation, might be the journal have the minimum requirements of the computational studies parameters that we have to.
We have several computational based study related journal like computational biology and chemistry, in silico research in biomedicine, then journal of biomolecular structure and dynamics, journal of computational chemistry and biology, 3 biotech. This always accepted the in silico work. So to publish in silico based work, just recommend for future that this is the lead compound and we need to validate in wet lab, including in vitro, in vivo, something like that will be accepted, inshallah.
Right. We have one questions more from Mr. Marlev. Marlev, would you like to directly ask Mr. Rooney, or I just read your questions?
Uh, yes, sir.
Yes, please.
Okay, good morning. Good morning, Mr. Rooney.
Yeah,
So
good
my name is
morning.
Marlef. My question is that when we perform like molecular dynamics and then we found the result, it's the docking is still not stable. What are the ways that we can do to improve the stable of the molecular docking? Thank you very much.
Yeah, thank you very much for your good question. If you find out that your compound is not stable, then increase the time of molecular dynamic simulation until get the stability. If you not get the stability after expanding the time, then you need to source again the compound from docking, or you can sense the protein to get the good stability. Sometimes we source the protein wrong. Now I will take a case study which I submitted yesterday. I choose a TLR4 protein to discover HMPV drug, but I chose unfortunately a wrong protein which did not show the stability. Then we increase the time of molecular dynamic simulation. Still, we didn't get the stable conformation. So when we choose the protein, then we should be careful. So which protein we will choose, in the protein data bank, we can see the amistrom. If the amistrom is less than 3, if the resolution of that protein is less than 3 angstrom, we will choose that one. That one obviously showed the stable complex, because before depositing that one, that PDB, that protein, they already perform the MD simulation for validation the structure of that protein. Thank you very much.
Thank you, Mr. Ronnie.
Thank you. Any more questions? What Mr. Malef asked was also my idea for asking you, Rooney, because it seems that doing MD is in the very last of our long sequence steps of doing computational studies. Then when we end up with something that we can proceed for our target, then it seems that we did for nothing in the initial steps. Or
Okay.
maybe can we publish, or we can yeah, we just through the previous steps without the MD data. Is it acceptable also?
Yeah, thank you very much. Uh, Here I have two types of virtual screening. One is ligand based and another is structure based. Suppose admit is ligand based, no need protein, only ligand. Then suppose I want to screen a database using ligand based, admit, then drug likeness only for ligand. After that, I will get some heat compounds. For that, I will go for docking with protein. So after docking MD simulation, there only one step, and usually when we choose the protein for docking, if we choose carefully, then in MD simulation it showed stability, because I already explained to Mr. Marlip that if we choose the best, if we choose the protein carefully, and when they deposit that one in protein data bank, they already performed the experiment, and after validate that model, they deposit that one. So we just need to choose. And when I explain in molecular dynamic simulation, I also said that if the apoprotein, I mean unbound protein, showed more higher than the complex, then also acceptable. Then no need to see about less than 2 or less than 3, no need. Just compare your result with apoprotein. That will be accepted.
Okay, right. So that there's a way, one way out for to sort out the problems there. Another one more questions from Mr. Putra. As I said, he's also doing computational chemistry. He published one of the docking compounds from base of our traditional medicine herbs here.
Inshallah.
Maybe you have experience about overcoming failures in PBSA calculations from MD simulation results in GROMACS. Have you ever done such experiments like that?
Sorry, I can't get you.
How to overcome failures in PBSA calculations from MD simulations results in GROMACS?
Okay, then we have another way is prime MMGBSA. After docking, you can suppose you perform docking for 10 compounds. So you can perform prime MMGBSA after docking. No need perform MD simulation, full analysis. Just after docking, perform MMGBSA. This is one kind of docking, but you will get the I mean correct complex to perform MD simulation. You can choose from that. So if you overcome the failure of if you want to overcome the failure when you perform MMGBSA using Grumax, Schrodinger, then another software, I would like to suggest you to perform prime MMGBSA before going to perform MD simulation.
Right. Any other questions from the audience? From Pa Fidel, from Pa David, maybe? Because our colleagues that are attending the seminar also doing for environmental studies, for might be we can modeling some molecules
Yeah.
in the purpose of environments. Any other questions? We almost half
I think
past
I'm
nine.
going to ask
Yeah,
one
Pa
or two
Fidel.
questions.
Good,
Okay.
Pa Fidel.
Thank you very much, Mr. Rooney. This is a very insightful talk, and this is also a very good thing to me because I haven't done this kind of thing before. So my question is related to something like this. If suppose we have passed, suppose we have a compound that has already passed all the compulsory step in computational based evaluation or something like that. Is there any chance that the compulsory step in the uh, wet lab evaluation that can be bypassed to follow up uh, that um, that research to I mean to come out with the drug, and in another condition, have you come across a condition that a potential compound has passed all the compulsory step or evaluation uh, based on the computer analysis, but when it comes to when it comes to wet lab evaluation, it uh, finally it failed. Have you come across a case like that? Thank you very much.
Thank you very much for your insightful question. And according to my supervisor, you know Dr. Muhammad Fadlizil Fasihi, computational approaches is until now is chance 50-50, 50%. That's mean now I would like to share with you a case study in Malaysia. Maybe Dr. Larry, you know Prof. Johnson from UKM. He get for a compound very good results in computational approach, but when he perform in silico, in wet lab, it's failed. Unfortunately, it's failed. But still now in the market, more than 100 drugs are available from computational study. Available and still now more than 100 drugs. So success rate is very high for computational approach. In the final state, sometimes it would be failed.
Okay.
Right. Thank you. Like what did we what we got through the COVID-19? Somebody says that the COVID vaccines comes from the just in silico studies, and then they just go through the the patients without the the clinical studies uh, because of the endemic situation right now, like that.
Yeah, um, that was the emerging issue. So we need the drug immediately.
Yeah.
Some antiviral drugs from in silico study, like remdesivir. You know, remdesivir is from in silico study. After prescribed to drug, after prescribed, then they try to perform validation. Because it was a world pandemic. So we need
Yeah,
drug
yeah.
immediately.
Yes, because the urgently
And fortunately,
situation.
remdesivir is work against COVID-19.
Yes, yes, remdesivir. Yeah, yeah.
Yeah.
Right. It's already 9:30, so it seems that we spent 1 hour of getting very, very interesting talk by Mr. Rooney. And I hope all of us, especially our students, could get something insightful for their studies because here we are also doing computational chemistry. We are studying also computational chemistry, and we would like to do more research about computational chemistry. Maybe some of you, our students, could contact Mr. Rooney for getting advice from his expertise in the in silico studies. But so I would like to thank Mr. Rooney. Thank you very much for your kindness, sharing us with your project, your knowledge about the computational chemistry, and hopefully your study will be successful immediately of doing the PhD under the Dr. Fasi's supervision. And
Inshallah,
I
pray for
hope
me.
we could meet again in the UMPSA Malaysia there for more talk about the another projects there.
Inshallah, inshallah.
Right?
And
So that I think, yeah, Mr. Rooney.
I would like to say something to your students that if you
Yes.
need any help in, especially in silico studies, just try to contact with me. I will always be there to help you, inshallah.
Thank you, Mr. Rooney. Thank you very much. I have Mr. Rooney's contact number so that anyone who wants to have or who wants to contact him, so just let me know and I will give you his number there. Or also the email address. Right. So thank you very much for your all of your kind attention. See you again in the next MRT session next month. And see you, Mr. Thank you, Mr. Rooney, and see you all there. Good morning for everybody and assalamu alaikum, Brother Rooney.
Walaikum assalam wa rahmatullah. And thank you very much for giving me this opportunity. And I am very happy to share with you about my research.
Thank you,
Okay,
Rooney.
assalamu
Thank you.
alaikum, everyone.
Waalaikum salam. Bye. Thank you. Good morning.
Thank you, Pak Reiner.
Thank you, Ibu Sinche.